Next, I would like to introduce Thierry Lave. Being involved early, I think, was a recurring theme already. And together with that panel, I think it only points towards doubling down on our efforts and starting as soon as we can. Thierry Lave.
Thank you very much, Ernst. And thank you for the invitation, and thanks again for the very important conversation we just had in the panel. So I'd just like to follow up on that and focus on this topic of inclusive research, thinking end-to-end about inclusive research and thinking, what is it that can be done early on, already in research and early development, to create the condition for the whole R&D to be more inclusive. And so what are the opportunities? I mean, we heard it from, it was very clear from the panel discussion, right? I mean, there are opportunities already for the patient. Everyone wants to have a drug that is working for him or for her. So having the right patient, the right medicine, at the right dose, at the right time is definitively something that is becoming increasingly important. Society, again, we just heard it very, very loud and clear. There are still a lot of disparities in terms of access to our healthcare, to our medicines, to our clinical trials. And we need data representative of the real-world patient population in order to optimize our clinical outcomes for all patients around the world. And then for our R&D organizations, there is an opportunity also for this inclusive research consideration to help also or to enable some of the drivers of R&D productivity. And this is what I'd like to cover here on this slide. If we think end-to-end about inclusive research, it's, again, creating the condition, or it's directly connected to this end-to-end evidence generation engine that we want to have within our R&D organizations. So being more inclusive and creating the condition for our R&D to be more inclusive of the different subpopulation is also an enabler of different drivers of R&D productivity. PTS, for example, so probability of success. If we have a better ability to predict the overall risk-benefit profile for compounds across different subpopulations, this is directly impacting the late-stage failures. So reducing late-stage failures, increasing PTS, obviously, which is also leading to a better cost efficiency. If we can reduce or prevent the costly late-stage failures through this better identification of population-specific issues. Contributing to speed as well, if we can have broader eligibility criteria to include different subpopulations or to understand under which conditions we could do that. This is contributing also to speed up development with faster patient recruitment. We all know that we still have also a significant number of post-marketing commitment, and again, doing this early investment into inclusive research already in early development will contribute to minimize those post-marketing commitment and overall create the condition to have a faster access to our medicines for all patient groups. Okay, so what if we think about early investment in research and early development? So what do we have in mind? So this is summarized here in a simple way on this slide. It's all about approaches that are existing already, that are innovative, yes, but are existing to a large extent, that will help us to identify and quantify variability drivers across patient subpopulations and, again, create the condition for this end-to-end approach. And everything starts by characterizing the drivers of variability, which, as you know, could come from a number of factors, intrinsic or extrinsic factors, and then leverage the possibilities we have now with access to real-world data, some emerging technology with organoids, organoid biobanks, and the possibility to integrate a lot of different information into this quantitative modeling and simulation platform, and of course, AI, increasingly AI, in order to simulate the impact of this variability drivers here on differential response across subpopulations, across patient population, or differences in drug behaviors in different subpopulations to enable, at the end of the day, the right drug at the right dose for all patients. Now, the first step is to characterize, to understand the sources of variability and deeply characterize this variability, this human diversity, by leveraging these advances in data science, including AI, machine learning, all other type of technologies, computational approaches, experimental approaches, really to integrate data from various sources, all kinds of assays, so genomics, transcriptomics, proteomics, data coming from experimental systems, increasingly in vitro systems that are more and more predictive of the patient, simulated data coming from different types of models, from AI, really to create the condition for this totality of evidence approach. That is an approach where we really try to integrate as much data as possible to create the condition for the clinical development to be more inclusive, and again, enable the right drug at the right dose for all patients. So that's step number one. So understanding, characterizing, quantifying variability, and then leveraging the advances we have in AI together with all the computational approaches, again, that are available since a number of years, to enhance our ability to predict drug response, drug behaviors in those diverse populations. And we are really at this inflection point where we can combine AI with those approaches that are already very well established to influence the whole value chain, helping identify the right pathway, right target, right molecule, right dose, and the right patient. Few examples taken from various groups. Here, one example from colleagues at Roche, where the team used a combination of AI or machine learning together with more traditional type of modeling to predict survival in patients with lung cancer with checkpoint inhibitors, and using also prognostic biomarkers to improve this prediction of individual survival in this patient. That is important, of course, then to tailor and to adapt treatments and enhance clinical decision making, and also to help optimize drug development. If we understand better who is going to respond, obviously this is helping to recruit better for our phase 3 trials and increase the probability of success in our phase 3 trials. So that's for efficacy. We know safety is also predicting safety more on a more individualized basis is very important. We know that the cost to society related to adverse events is still very, very significant, several hundreds of billions every year. And it's good that there are lots of effort to move into more predictive safety, more individualized, more personalized safety, using also a combination of mechanistic models that are already well established, like here, for example, in the space of cardiac safety. These are examples taken from some of our colleagues at Roche as well. So combining mechanistic safety models with AI type of models to try to identify those risk factors for different parts of the population that help to predict the risk of cardiac arrhythmia for different subpopulations. And there are lots of efforts going in this direction of improving our ability to predict safety for different types of organ safety. This is again cardiac safety, but there are efforts in other type of organ toxicities, organ safety, efforts that are in collaborations, the result of collaboration between industry, academia, and regulators. Now, here's some work from our Genentech colleagues on the use also of real-world data and AI to help designing more inclusive clinical trials. In this case, for oncology trials. And again, we heard some numbers earlier in the panel discussion showing that our eligibility criteria might be too strict and represent a significant barrier to deliver patient benefit. These are some numbers cited in these papers, where only 8% of the US cancer patients enter clinical studies, 80% of the lung cancer patients do not meet eligibility criteria, and 86% 86% of clinical trials failed to recruit within the timeline, so which significant impact on the speed of research and development. And what the colleagues did here is to use this AI together with the real-world data to try to revise the eligibility criteria and come up with so-called data-driven eligibility criteria, and showing that patients who would be excluded from clinical trials could actually benefit from the treatment and should be considered for enrollment. So using that data-driven eligibility criteria, they could double the number of eligible patients without a lot of impact on the overall survival hazard ratio, right? So, which means that people who are excluded would actually benefit from the treatment. And the other observation made in the paper, it's not on the slide, but there was, it didn't have, this data-driven eligibility criteria would not have significant impact on the withdrawal of the clinical study because of adverse events. So patients who were excluded would actually not experience more adverse events than the one who were included based on the original eligibility criteria. So more diverse patient population included as a result of this data-driven criteria, with a significant impact on including more female patients and more patients over the age of 75. Okay, another consideration now, if we think about starting early with this kind of inclusive research consideration to create the condition, again, for the whole R&D to be more inclusive, it's important that the assays that are being used early in research are also representative of the diverse population. So it's true whether these are computational systems, AI, or experimental systems. And here, I just wanted to highlight what is happening for organoids, for example, the creation of organoid biobanks, with efforts in developing this human model system to represent the more diverse real-world populations. And these are some examples of papers and work from different groups around the world working really in this direction, which is very encouraging to see, with this diversity on the dish effort here, where the team is leveraging organoids to reflect different sources of variability, genetic ancestries, sex differences in health and disease. Other groups here at Genetech working towards inclusivity already in preclinical drug development, again with organoids, and they do some work using intestinal organoids to contribute again to this improvement in drug efficacy and safety assessments for compounds, to advance precision medicine, to help better understand disease mechanism, trying to contribute to reduce health disparities and improve our clinical trial success rate. Other publications also with this village on a dish approach that has been published as well. So all is very encouraging. Of course, it comes with a number of challenges, as you can see here. Of course, you need to acquire sufficiently diverse biological samples from different donors, from a broad donor population, including underrepresented patient groups. I mean, there are some challenges related to ethical consideration, data privacy that need to be addressed, more technical challenges related to the scalability, the automation of the assays that are being used, and the application of fair data principles also to this kind of technologies. So one of the last slide is to show the impact of this early investment. So including this consideration already in research and early development, what is the influence on later phases and on post-launch activities? The investment in research and early development can contribute to identify the causes of variability across different subpopulations, provide initial dosing recommendation in different parts of the population, including underrepresented groups. This work is also important to identify variability factors, whether they are significant or non-significant. It is important to find out, and can help also to identify variability factors that are going beyond the traditional race, ethnicity, age, and sex. And then that has an impact on, of course, enabling more inclusive late-stage trials, lowering the barriers for patient participation into our clinical studies, better stratification of our assays, fewer biases in the recruitment for our clinical studies, impact on post-launch activities as well, contributing to improve outcomes for representative patient population, better dosing guidelines, contribute to more immediate access to medications to a broader population, and at the end also enhance efficacy and safety for representative patient populations. And and that's the last slide. So really this investment in early research and early development is important. It is helping us to move from an approach where we are a bit reactive, focusing more on the percentage of demographics to be included in late-stage studies, to being more strategic and proactive in driving this inclusive research topic from the early stages of drug development, thinking about all the populations our medicines might serve to ensure favorable risk-benefit profile across the different population. This is a huge ambition. Of course, there are some challenges to be addressed. I mean, investing early has a cost, but all the tools that we have, all the innovations that we can leverage will contribute to lower the barriers to be as inclusive as possible. And I stop here. Thanks.